Good morning, good afternoon, good evening, and good night. Welcome to the Big Fat Conversation, broadcast live in 4 different continents with guests in 4 different time zones and 2 hemispheres. For those of you who are new to UK Low Carb, I'm Dan Grief, your host, and this is a podcast where we share the N=1 stories of people all across the world who have improved their lives by following the low carb and keto way of life. The aim of the show is to build a grassroots movement for change so that we can take on the current nutrition status quo and liberate millions of people from the misery of metabolic syndrome. And if you don't already, please subscribe to us on YouTube. The channel is UK Low Carb, and also our Facebook group, which is UK Low Carb, so that we can help spread the word. Tonight, we have a very, very special show, and that's because I'm joined by a very special panel of experts, and they are as follows. We have Cynthia Thurlow, Amber O'Hearn, Richard Morris, and Nina Teicholz. Hi, guys.
Hello.
Good evening.
Great to have you here, and thank you so much for joining me. Now, just to go through the process for this evening, we are going to go through a number of articles. So each of my panelists has brought with them a paper or an article to share. The idea is the panelists will then introduce the article to us, and then they'll have an opportunity after that to have a conversation. So we're going to start off firstly with Amber. And Amber has a very interesting paper. Now, I'm sure many of you know Amber O'Hearn already. She's a data scientist and academic writer. She's been completely plant-free and a carnivore for the number of years now. She's written publications in several fields, including theoretical mathematics, cognitive psychological, computational linguistics, and more recently, evolutionary nutrition and biology. I'm so sorry, Amber. I tried my hardest there, but I knew I was going to trip over some of it. So you have an amazing paper for us today, don't you? And that one is the one here. Origins of the human predatory pattern, the transition to large animal exploitation by early hominins. And I'm just going to pass you over now for your 15 minutes. Thank you very much.
Thank you. Yeah, I really love this paper. And obviously, I'm not one of the authors, but I have seen the lead author, Jessica Thompson, present on it a couple different times and had some brief correspondence with her. So I hope that my interpretation will be true to what they were trying to bring here. I came across the paper because I'm deeply interested in humans and what makes us different from other animals. Of course, the main difference, or at least my favorite difference, has to do with our brains. And it's not just brain size, but structure and use. But another aspect of humans as a species that makes us very different from other primates and from other mammals, generally most of them, is our relationship to fat. So on the one hand, we have an extraordinary amount of body fat for primates. Stephen Cunnane dived into this quite a lot and talked about its connection, in fact, to the brain. And the other, another aspect of our unique relationship to fat is how easily and quickly and deeply that we can engage into a ketogenic metabolism. And I think that that's intimately related to our body fat and intimately related to our large brains and meeting their needs. And so it's in the course of my interest in exploring what makes us different as humans, which includes our evolution, obviously, that I came across the paper. Now, the paper's interesting in and of itself for reasons that I'll get into, but it's a hypothesis paper, and it's a hypothesis about how humans became so carnivorous and carnivorous in the unique way that we are, which they will explain, or I will explain on their behalf. But there's an observation that's brought up that's in the formulation of this hypothesis that has implications for human physiology and our relationship to fat. And that's one of the reasons that I find this paper so compelling. It's structured very nicely. So what the authors do is they present a problem that they see in the literature and anthropology, and they propose a solution, and then they, of course, as good scientists as they are, they suggest ways that that hypothesis could be tested. So we understand that meat eating is an integral part of what it means to be human, what made us human. It allowed our brain expansion, and it shaped our digestive system, and it influenced the way that we interact socially, and all of those things are really important for humans and defining what humans are. But one thing that kind of gets glossed over when we talk about human meat eating is that it's really different from the way that other primates, extant primates, so chimps, for example, have a relationship to animal sourced food. So animal sourced food is important for many different animals, even those that aren't primarily carnivores. And chimps, who are our closest primate relatives, they do make use of animal source food regularly, but it's a small amount really of their calories, and it's not everyone who gets that much. The alpha strongest species will get more opportunity for that kind of food than others. But what they don't do is hunt large animals. So the carnivore pattern, what's termed the carnivore pattern of animal eating, is to eat animals that are at least as large as yourself. And that's kind of a heuristic between thinking about animal sourced food as something that a primarily herbivorous animal would have as an addition, an enhancement to their diet, and an animal that's truly carnivorous. It might seem a bit arbitrary to talk about the prey size, but the prey size distinction is actually really important for a couple of reasons. And one of the reasons that it's important actually has to do with fat, because generally speaking, the smaller the animal, the leaner it is. So if you get a larger animal, not only have you gotten more food all at once, but pound per pound, you're going to get a higher proportion of fat. And that's important because calories is is arguably the most important thing that you're looking for when you're looking for food. So one of the things that is pervasive in a lot of the paleoanthropological literature is a kind of latent assumption, maybe not very well examined, that large prey hunting in humans, for example, is just really an augmentation or an enhancement or a gradual growth of the small prey hunting that you would see, for example, in chimps and bonobos. A lot of people look to chimps and bonobos and use that as a kind of model of our common ancestors, and that's actually a mistake, because if you look at two species that have a common ancestor, each of their evolutionary lines happened. They're both evolution, right? So it's a mistake to think of chimps as representing the common ancestor between us and chimps. So the behavior that we see in them today, we can sometimes use that to think about what the behavior might have been in our common ancestors. But in some cases, that kind of behavior that we would see that would differentiate us could equally be something that they derived and evolved too that wasn't present in a common ancestor. And so if you're looking at the difference between small prey hunting and large prey hunting, it turns out that there's a whole suite of behaviors, a whole set of behaviors that are different when you're approaching hunting large game than when you're approaching hunting small game. And it turns out that it's not even really necessary. The authors point out that it's not even, perhaps not even likely that our common ancestors had the kind of small game hunting behavior that chimps do now. And so to assume that our large game hunting is just an expansion of that small game hunting is possibly a fallacy, really. So when we when we ask the question, why do animals eat what they eat? What does an animal's diet consist of, and why do they choose the certain foods that they do? One lens that we can look at that through is called optimal foraging theory. And basically, it's just a way to try to figure out one question that it would ask, for example, is what would you always go for if the opportunity arose? So you have all these foraging opportunities, and some of them might go by and the animal doesn't go for it. But some opportunities come up and they will always or almost always go for it. So what makes the difference between those kinds of opportunities? And the way that they do that in optimal foraging theory is really just a kind of economic viewpoint. So it's a cost benefit analysis, and the idea is that the kind of foraging that you will always go for is the one that will provide a lot of benefit for a small cost, right? So benefits, when we're talking about foraging theory, are what the food is going to bring to you nutritionally. Micronutrients, of course, are important, and protein is important, but far and away the currency that we're talking about when we're looking at how animals choose what they're going to go after is energy. So that's calories, right? And this is important because when you look at a large animal compared to a small animal, and the main difference between those is fat, that means that the main difference between those is actually energy. So the benefit of being able to get a large carcass is much more than just the amount that comes along with it, right? And this means that it doesn't matter how abundant those smaller animals are. If you think that you can get, if the opportunity cost, or if the cost is low and the opportunity is there to try to get something larger, you will divert resources even from something that's abundantly available. So we might compare this, for example, to dating. So if you are looking for a mate and there are a lot of people in the room who are interested in you, but you're not interested in them, it doesn't really matter how many of those that there are if you think that you have a good chance of getting the person that you prefer. So the abundance of smaller prey likewise is kind of irrelevant once the opportunity arises for you to get larger animal if there's a good chance that you can get it. So the chance of getting it comes down to the costs. So when we talk about costs, the cost difference between large prey and small prey, one distinction that we can make is between getting the entire carcass, which we would do in a hunting situation, and the scavenging situation in which we are getting primarily the bones. So the a term that they bring out in this paper is inside bone nutrition as opposed to outside bone nutrition. So inside the bone, we have marrow and we have brains. And these are very, very high in fat and also lots of good nutrition. It's not just fat, but it's even higher in fat than the muscle meat that would have been attached. So now if we're comparing the costs of different kinds of foraging strategies, we can look at scavenging, we can look at hunting large game, and we can look at hunting small game. So one of the costs that's very different between small game and large game is exposure to other large carnivores. So we've exposure to large carnivores in an open habitat hunting situation or scavenging situation is very dangerous to the hunter, of course, and it also provides a kind of competition. So the exposure cost for scavenging is actually quite a bit lower than the exposure cost of hunting, because you're not directly competing with the other large carnivores who are there, and you can wait until they're gone, so there's less exposure for your own safety. And another way that scavenging this inside bone nutrition has a lower risk cost associated with it is because of bacteria. So when we're scavenging, obviously the meat that's been left, if there's any meat that is left, has had some exposure to bacteria, other animals, species even. So that can be a big risk of infection. And it's believed that one of the reasons that chimpanzees don't scavenge, which is very interesting actually, they will pass up opportunities to scavenge. So it's right there and they don't take it. And one proposed reason for that is because of the exposure to bacteria. But when you look at the inside bone nutrition, if you were able, when you were scavenging, to actually get inside the bones and get into that fat, it has a better shelf life, if you will. It would last a few more days before it would start to rot. When it's inside, it's protected. So the ability to be able to get inside bone nutrition suddenly is a way that reduces the cost, and it actually increases the benefit of interacting with large carcass animals compared to small ones. So we would be able to do that, and we know that there was this transition from not being a large game hunter to being a large game hunter. And so one of the ways of trying to figure out what happened to originate that is to think about it from this optimal foraging theory point of view, where you're saying, well, at what point did the cost become low enough that we could take advantage of those benefits? One of the things that lowers the cost is having the available technology to get into that inside bone nutrition. And this is really one of the cruxes of the paper, where the authors talk about percussive stone tools. So one of the other assumptions that we have when we're talking about the human hunting pattern is we have meat and we have the idea of flaked stone tools. And so flaked stone tools they're really an important part of the butchering process, and the ability for us to get the meat when we have a large carcass to break it into small enough pieces to be able to deal with, to get it off the bone. It's very tough. And so that kind of tool has always been associated with our meat eating. However, if the exploitation of large game had to start with flake stone tools, then we kind of have a lot going on at once that had to happen all together, and it becomes a kind of bootstrapping problem, because flake stone tools are actually a secondary tool. You need to already have a tool in order to make that tool. And so if you're proposing that we suddenly went from not hunting to hunting and requiring us to have those tools, we get into like a problem of how did we figure out how to jump that 2-level tool technology problem in order to do something that we weren't doing before. And then if you bring the brain into it, so some people might say, oh, well, we suddenly had the brains, because of our brain expansion, we were able to figure out how to make this tool that would allow us to eat meat. But there's a circularity in that, because we know that the expansion, or we believe that the expansion of the brain required the meat to be able to do. So now you're saying that you needed the tool to have the brain expansion that you needed to make the tool. So this is a kind of problem that they've set up. A percussive tool is something that we almost certainly already had. Chimps have it now. A lot of primates have this ability, or this ability to use percussion tools. And what a percussion tool means is simply you're banging something, right? So you need to have the ability to hold it in your hand, and then you're just smashing. And other primates already have this ability and use it to get into food in the form of nuts or shells of small animals. That's something that we likely already had. So the transfer of this knowledge using a percussion tool into the scavenging situation, the authors are suggesting form this bridge to allow us to go from not using large animals to using large animals. So we took the percussive tool, used it to break into the inside bone nutrition and get that, and this was the beginning of our introduction to large game and all of the benefits that it brought. There's one little point that I think is really it's evocative. When you use a percussion tool, when you use a stone tool, it begets flakes. They come off it as a product of what you're doing with the banging. Now, monkeys that are known to use stones to break into, say, nuts, they completely ignore these flakes that are going off it. But you can kind of imagine this scenario where over and over again, humans are breaking into bones, and the bones might have a little bit of flesh on it still, but they're mostly interested in the fat that's inside. And little flakes are coming off, and somebody eventually notices that that flake could actually be useful to get more of the meat that may be attached to the bones, and that this somehow progresses into our use of butchery. Now that's, it's a kind of a fanciful imagination, but I don't think it's actually implausible. So I think this is a really interesting hypothesis that solves a problem that we didn't necessarily think about, the fact that we have it. They have suggested that this focus on the flaked stone tools has caused us to sort of find what we're looking for, because if we're looking in those old sites for evidence of the particular kind of knife marks on bones, for example, we may overlook the kinds of evidence for percussive stone tool use that they are proposing. And so they have suggested different ways that we could start looking for that evidence and see if the hypothesis is supported. But one of the things that I think is really important about the whole that it brings to light, and it's not really a hypothesis, because we do have evidence that we did break into marrow and into brains, and that is that fat could be considered actually the gateway to humans' carnivory, not actually small animal hunting, but fat itself. And it suggests that our relationship to animal source food, that the fat part of that is actually an older and more strongly evolutionarily, a longer evolutionary relationship than the more proteiny parts that we tend to think about when we think of meat eating. A lot of people will actually call it your protein, but I like to think of it as something much more than that. And so that's why I think this is important.
Fantastic. Thank you so much, Amber, for that paper. Just to let everyone know, by the way, I'm just tweeting out the link. I've actually got a website with all the links there. I'll see if I can put it on the bottom of the screen as well to let you know. And there's a website with all 4 articles today, so you can go and check that out for yourselves. Before I open up to everyone else to discuss that paper with you and do a little bit of Q&A if that's possible, Amber, as well, just to let you know, if you are following this on Twitter tonight, you can, this morning, this afternoon, wherever you might be, then you can, of course, follow the hashtag BigFatConvo. I've put it on the bottom of the screen as well. And I've actually been trying to tweet while also listening and making notes. So I'm multitasking, which is very unlike me. Okay, I just want to open up to everybody in that case to have that chance to have a discussion. And I'm going to hog, if I can, very quickly and just ask something, if I may, Amber. you said there about the fact that, you know, it could, and my degree's in archaeology, so I kind of find that human evolution is a very interesting problem to have, in the fact that you're looking at quite often the material remains of bone and stone. You're not seeing flesh, because of course the flesh went millennia ago, and so you're trying to, you know, make an assumption in some ways based on the evidence you do have, which makes it quite hard. Do you think then, I know you said what the paper thinks, but do you honestly think that the fat was the gateway that got our brains to grow? Because I was reading earlier on in your paper about the fact that Australopithecus afarensis, of course, was the first kind of biped with the human hand, with a grip and everything else. So do you think that it was the fat that made all those things happen, or something else which made us eat the fat to then make our brain grow?
I think it's quite plausible. I mean, one of the statements that they make in the paper is that even Australopithecus had undergone a brain expansion that was comparable to later brain expansions that we associate so much with what makes us human. And so obviously at that time, the hunting was not as much as it was later, and our meat eating was not at the apex that it later became. And so if they were to have had the influence of fat at that time, that it does neatly explain how that could have happened.
Yeah, excellent. Okay, so over to the panel. If you would like to speak, just unmute yourself and go for it. Nina, over to you.
Yeah, thanks. Amber, I just wanted to understand something that I think you said, but I'm not quite sure. Is it the commonly held belief that the first kind of hunting that humans or early humans did was small animal species, and that this paper suggests that instead it was large animals? Is that part of what makes this paper sort of one of the hypotheses, and specifically in this paper?
Yes. So,
am I understanding?
Yeah. So the common thing to do is to make an analogy between the pan species, chimps and bonobos, and how they incorporate animals' foods into their diet through small game hunting, and to assume that that was what our earliest hominin ancestors were doing, and that they kind of gradually expanded out. And I don't think that it's ever been explained very well, except as, you know, just intuitively, naturally, we started having, as we had better tools, we had the ability to get larger and larger animals. And so they're suggesting that actually these two kinds of behaviors are completely unrelated, and that you don't need to have smaller game hunting as some kind of precursor or early form of hunting, and that in fact they're completely different behaviors.
Excellent. Okay, so would anyone else like to make a point, a comment, or ask a question to Amber before we move on to our next speaker? Okay, excellent stuff. Just one thing I want to say about evolution, which I think is really fascinating, is that people quite often talk about evolution, and this is where people, of course, haven't studied the subject, but they almost talk about evolution like we have evolved to this point and chimps and bonobos have evolved to that point. And it's not true. We've all evolved to the same point. We've just taken slightly different paths, and we have a common ancestor, which is, you know, links us all together, but we've actually evolved to our own environment. So to look at like a chimp or bonobo and say, you know, they are less evolved is just wrong, isn't it? And that's why the comparison sometimes is actually completely wrong, because the environment they evolved in is different to us. That's why they're different to us in the first place.
Yeah, and if I could just elaborate on that, or make a kind of an analogy. So the fallacy in thinking of chimps as a way to model our pre-human ancestors, I mean, there are it's not completely wrong. We can definitely make some inferences, but the idea that they are always a good comparator is comparable to me to the idea that modern hunter-gatherers are kind of a recapitulation or a less evolved form of humans that we can compare to early humans, or even early modern humans. They didn't necessarily live or eat the way that modern hunter-gatherers do, any more than chimps represent our common ancestor.
Yeah, fantastic. So ethnoarchaeology is actually quite limited, definitely. Okay, thank you so much, Amber, for that. That was a fantastic way to start the show. And before we go any further, I just want to share some of the comments we've been receiving from people around the world. We've got a lovely one here from Kylie, who says hello from Australia. Actually, that reminds me, I should just say, Gary Fettke was going to be on the panel today. And unfortunately, because of his operation he had earlier this year, he's still recovering. So if you are watching Gary in Australia, just hello. And I'm sorry you couldn't be here, but we miss you. I hope you get well soon. And Kylie, thank you for watching. Really appreciate that. There's so many comments here from a good friend of mine, Nicola Howard. I want to share something that's really wonderful. I'm going to share this one. So given that Homo sapiens brain is mostly fat, that would then seek larger, fattier prey makes sense. Yeah, exactly. So the fact we'd seek those prey eventually would make sense to feed those brains. But which way round it goes, I don't know. And just to let you know, Nicola, you asked there, will we actually have the link to the show notes on the podcast? We will next week. So you can read all these articles on there as well. Okay, right. So we're going to move over to our next speaker in a moment. And that, of course, is Cynthia Thurlow. So just to tell you a little bit about what she's talking about today, and also to mention a bit about Cynthia as well, if you don't know. So Cynthia Thurlow, I'm sure you know her already, but she's a nurse practitioner, entrepreneur, and host of the Everyday Wellness podcast. She's a published author and famed TED Talk speaker, and she has a book coming out soon, haven't you, Cynthia? I'm sure you can tell us about that in a moment. And just to let you know, her paper is called Remodeling of the Gut Microbiome During Ramadan, Associated Intermittent Fasting. So I'm just going to hand over to you now, and the floor is all yours.
Thank you. It's so nice to be with everyone this evening. And Amber, I loved hearing your presentation. I have a lot of ideas ruminating in my head, and I'll definitely reach out to you afterwards as I kind of formulate questions. Not surprisingly, I asked Dan if I could present a paper about intermittent fasting. I'm getting ready for a talk next week in Scottsdale, and it just seemed incredibly timely. And so this paper specifically is talking about the net impact on the gut microbiome during Ramadan. And what I found most interesting was that the objective was really to look at establish the effectiveness of 1 month of intermittent fasting on the gut microbiome. And, what I found interesting was they had 2 cohorts, and this was done over 2 years. And for anyone that's, listening, I think intermittent fasting for many people has become a sustainable strategy to try to harness, metabolic flexibility. And when you look at the statistics, not just in the United States, but in most westernized nations about the ill health of so many of us, I think there was a UNC study that was done a few years ago that documented that only 12% of US adults are metabolically healthy, meaning people that are within a healthy weight, have lower triglycerides, healthy blood sugar, as well as a few other biomarkers. And so I think it's really relevant to be talking about intermittent fasting. And, certainly for individuals that embrace fasting as part of their religion. Obviously with Ramadan, this is something that they do on a yearly basis. It was really relevant and timely to look at this. And so they looked at these 2 cohorts over 2 years, and they observed that there was significant changes to the gut microbiome. And they actually had one cohort was young and healthy without metabolic disease. And then there was a little bit of an older cohort that some of them could have indeed been overweight and had some degree of insulin resistance. And so the consistency that they were seeing was, during this 16-hour fast that they were doing every day over the course of a month, that they were getting increased levels of butyric acid. And for anyone that's familiarized with what this is, it's a short-chain fatty acid that provides cellular energy in the colon. And we know that it provides about 70% of the colonic energy needs. So really significant. It helps, butyric acid butyric acid is involved in breaking down bacteria, and fiber can be one of the elements that contributes to this. And I'm sure that Amber probably has some very strong opinions about whether or not fiber is something that we really need in our diets. But we can also get butyric acid supportive foods beyond just fiber. So when we're looking at this paper, the things that I found really of significance, primarily was the fact that we know, when we're really looking at the research on intermittent fasting, we know that there can be significant impact on circadian biology. We actually have sensors throughout our body, not just in our brain, in terms of how our gut empties and how our body responds to food and food cues. We know that we can get remodeling the gut microbiome depending on, timing of eating. And the information provided in this study, although not statistically significant in terms of sample size, that's one of the things I always talk to my female patients about, that the sample sizes are relatively small. One was 30 volunteers, another was 37. It at least gives us some information that there can be studies subsequent to this that might be able to dive a little bit deeper into the science. And so we're really looking at these young male cohort that was predominantly out of China. They had 30 volunteers. There was a middle-aged cohort, and I don't believe it defines the age, but as someone who's middle-aged, I would imagine they're probably at least in their 40s, and there were 37 healthy volunteers. And over the course of this, this 1 month period of Ramadan, the primary outcome in the gut microbiome was ascertained by fecal DNA. So they were able to actually look at DNA samples and be able to extract the phyla of microbiota that was going on there. It was interesting that they mentioned the p-value was less than 0.05, which they found to be statistically significant. They mention here that, sorry, I'm just kind of skipping around. And because I have my blue blockers on, I can't see the paper as closely as I would normally. I'm trying not to make sure that when I go to bed later, I can actually fall asleep from being under these bright lights. But the net impact was that when they looked at the DNA-based sequencing on the stool samples, that they were seeing more butyric acid producing bacteria. Again, butyric acid provides energy to the colon and is really significant. Again, we can go back and talk about that. There's a lot of people who believe fervently this is related to fiber intake. Butyric acid sourced foods can come from some protein-based foods as well. But it's interesting that they mention here, we did detect there was a positive correlation between body mass index and the abundance of bacteria specific to this particular type of bacteria that they found. And what I think is really relevant is that although, again, the sample size was not, more than 100, and that was something that was always kind of hampered on me in graduate school, I think it really brings to light that there's more to fasting than just body composition changes. The gut microbiome is really so largely impacted by what goes on with our health, the interrelationship between gut and brain, mood, neurotransmitters, et cetera. But, the other piece of this that they talked about was that associated also with this increase of butyric acid was that you got improvement in not only blood glucose, but also body mass index and body composition, which for many people is the primary driver for why they are interested in intermittent fasting. But I just found this to be an interesting and relevant, paper that I think for a lot of people that are focused on intermittent fasting, it provides another context of another population of people that can benefit from eating less often. That's really what it comes down to. Giving more digestive rest, giving our bodies more time to, have time to digest food and, really take advantage of not eating quite so frequently.
Fantastic. Thank you so much for that, Cynthia. And I thought that was a really fascinating topic to have as a paper. I think the thing that I found really interesting when reading this was the fact that, like you said, they were based in China. And I just wondered the cultural difference. So, my wife's family are Muslim, and at Ramadan, I know many of their family would actually have, a big celebration at nighttime, and they'll eat lots and lots in the nighttime to really fill up, and it'll become like a party. Now, some people I know lose weight, as some of these people did. Other people put weight on, depending on how they're eating. And the other thing I was thinking as well is what you said about circadian rhythm, because as we know, Ramadan changes throughout the year. And actually, I wonder what the difference in a January Ramadan compared to a July Ramadan would be based on the light that people are exposed to, and how does that affect things? I have no answers, but I think it really opens up a lot of questions, which I think, I'd love to know. I was going to open up to everyone else now, though, as well. And now it's your opportunity, if you so wish, to ask questions, make comments, or anything you wish to say.
So I might make one comment. The pathway of butyric acid is via beta hydroxybutyrate and acetoacetate. So it is arguable that feeding your colon 70% of its substrate by converting fiber into converting indigestible carbohydrates into butyric acid is other short chain fatty acids is one way. Another way is that's via the luminal supply to the cells, but the other way is basolaterally via the blood supply if you've got beta hydroxybutyrate in the blood. So, for ketogenic people who are ketogenic, they always have some level of acetoacetate and beta hydroxybutyrate in the blood. So, that's potentially it'd be good to see bowel cancer experiments controlling for ketogenic dieting.
I think that's such a good point, because for people that are familiarized with a ketogenic diet, low carb, intermittent fasting, a lot of the brain benefits is the crossing of the blood-brain barrier with the BHB, beta hydroxybutyrate. It's a lot of the reason why people feel all that mental clarity. So that's certainly an excellent point, and definitely one worth looking into. Thank you.
Fantastic. Amber, what do you want to say?
I just think it's really interesting, because we know that what foods that you eat can have a big influence on which kinds of bacteria will be more dominant than others, and the resulting nutritional benefits of that. But what's really fascinating here is that the effect that we're seeing of the butyrate, which is an energy source that the colon is using, is actually coming from not eating. So it's one more factor that you might not think of if you're just thinking about, what are the types of foods that we can use, but actually the timing and the rest from food that can actually have a positive difference is really perhaps unexpected.
Well, actually, Amber, just thinking, we're almost like an example of that in a way, because we are completely different times of day, although we're actually talking at exactly the same time of day. And it'd be very interesting to see, if we had something to eat now, what would Richard's response be in terms of insulin release and his microbiome and everything else, compared to Cynthia, compared to yourself, who's like in the sort of early evening, late afternoon, compared to me, it's now 10 to 1 in the morning. So, we all have, it's interesting, an experiment to have that, I think. I'd love to know for sure, because I think it's, like you say, it's the time, not just the not eating, it's the time. Would anyone else like to make a point or anything to Cynthia's paper, or say anything at all? No, fantastic. Well, in that case, we're going to move on. Before I do, I have a question here by somebody who just left a comment, and I'm just going to put it on the screen for you. And then if anyone wants to answer this, they can. But because it doesn't directly relate to any of the topics, I thought it'd be good to kind of cover now. So Helen says the following: "Thank you for taking time out of your busy schedules." It's okay, I was only sleeping at this time anyway. I'm not that busy. I'm wondering if anyone has an opinion about cured meat versus uncured meat from the nutrition, bioavailability, and or consumption safety perspective. That's a good question, actually. Would anyone like to have a go at that one at all?
I think Nina might have something about it later on.
Yeah. Do you want to go? Do you want to talk about that for now, Nina?
I'll just give a quick answer that there is a new paper out in the journal Nutrient. We can put a link to that maybe, that looks at processed meat, which is not specifically cured meat, but processed meat is what was named by the WHO as a probable source of colorectal cancer a few years back. And this paper that just came out is fantastic, because it's the first paper that really looks at all the studies that have been used to basically to accuse processed meat of causing cancer. And it evaluates these studies according to a rigorous system called the GRADE system. And it evaluates 130 papers, many of which were used by the WHO, the WHO, to make this decision. And it ultimately concludes that that only 20 of these papers were actually on cancer and were of good enough methodological quality, and that the result, and those papers show either no effect of processed meats on cancer, colorectal cancer, or very, very tiny effect, marginal effect. So it's a really good paper because it sorts through the evidence to say, looking at the best evidence, this is what the best evidence says. So I know it's not quite on the topic of cured meat, but I think that's a great paper and a really important contribution to the literature.
Yeah, fantastic. Thank you for that, Nina. You always hear that, don't you, with that argument from the plant-based side. And it's interesting to get the evidence now to say that's not the case. So excellent stuff. Right. Okay. In that case, thank you so much, Cynthia, for your paper. That's fantastic. And we're now going to move on to our next speaker now, which is a certain Mr. Richard Morris. So what can I say about him in polite company? He's an official keto dude, which is, I think we all know that for sure. He's a co-host of the Two Keto Dudes podcast. In 2014, he reversed his diabetes with a ketogenic diet, and he's just completed his BSc in biochemistry, where he actually graduated with honours. He's also some big, exciting plans ahead of him in the career of academia. Do you want to tell us a bit more about that, Richard?
Sure. I was a software developer for many years. I worked on Wall Street in the turn of the millennium, and I retired from software developing at 40 because I got a diagnosis of prediabetes, and I wanted to work out what the heck that was all about, and keeping my feet and my toes and lower extremities seemed to me more important than earning a salary. So I managed to glom onto a video by Tim Noakes talking about ketogenic diets, and I jumped in, and all of a sudden I discovered I had an interest in biochemistry. I didn't realize I was interested in biochemistry part of that. So I started the podcast. Carl and I both turned out type 2 diabetes around. We've run multiple conferences, and we get roughly 250,000 downloads a month of people who have all managed to turn their own diabetes around and are trying to help their own family members and the like. So it's been quite an adventure. But I ended up getting a BSc. I ended up deciding I want a platform to be able to not only learn more about the biochemistry of type 2 diabetes and ketogenic nutrition, but also to get a platform really to talk about it. So I'm in the process of doing a master's in bioinformatics, and then I'll do a PhD. I did an honours in computational chemistry, the undergraduate, and in biochemistry, and I've published one peer-reviewed paper, peer-reviewed study on the epithelial membranes, and I'm in the process of finishing up a comp chem study into the inner mitochondrial membrane in response to fatty acids in the diet. So that's a potted history.
Brilliant. You know what I love there? You took your passion, which came from a necessity to try and save your toes, which obviously was like survival at first. It became your passion project, and now it's become your qualification. And when you're a professor, Morris, I'm sure will
I will never be a professor. I don't want tenure. I'm more than happy to dabble.
You don't fancy university
experimentally? I do not. No.
Fair enough. Well, let's go for yours. Now, this is slightly different because you actually have a video. So let's see if I can just add that to the stream.
I'm going to share a fresh study that was published on the 29th of September 2021 out of Professor David Ludwig's lab at the Harvard Medical School. The study, Wong et al. 2021, tests the hypothesis that insulin secretion predicts body composition following weight loss. The study is a reanalysis of the results from two different weight loss studies managed by Professor Ludwig's lab to try to explain why different people have completely different results from calorie-restricted weight loss. Most people who diet to lose weight through energy restriction only really want to lose body fat, not lean tissue. Lean tissue is more metabolically active, it burns more calories, so if you eat fewer calories in and end up losing lean tissue, then you will have fewer calories out, and you'll have a harder time keeping the weight off. Energy results from two different weight loss.
Hello, Richard. I don't know if you can hear that.
Studies managed by Professor Ludwig's lab to try to explain why different people have completely different results from calorie-restricted weight loss. Most people who diet to lose weight through energy restriction only really want to lose body fat, not lean tissue. Lean tissue is more metabolically active, it burns more calories, so if you eat fewer calories in and end up losing lean tissue, then you will have fewer calories out and you'll have a harder time keeping the weight off. Energy-restricted diets lead to reductions in both lean and fat mass, but it seems that the ratio between the two varies a lot from one person to another.
I'm sorry, Richard, I believe
State
it's...
food study from 2018. We'll call this FS2.
Okay, I'm really sorry,
It
Richard. I
looks like
think
it's not
that's
working.
jamming up. It might be because we're trying to play it from Canberra to Cambridge. You know, so that could be some of it. So do you want to just go through this for us, if you don't mind, and
Sure.
just tell us a bit about it, okay?
So let's see if I can get... Unfortunately, I have some fairly detailed figures that I'll need to be able to show. But essentially, I'm bringing up a paper that's only been published a couple of weeks ago by Ludwig's team, and it's a reanalysis of two existing dietary studies, and one of those studies we know quite well, and one of them hasn't even been published yet. So the one that we know well is the study that was funded by Gary Taubes and Peter Attier and Nusi. And that study observed that people who lose weight and keep that weight off have burn 52 calories a day more for every 10% reduction in carbohydrates. And what that really means is that if you think about calories in, calories out, if you calorie restrict and you reduce the calories coming in, and your body automatically reduces the calories coming out at the same time, then the net effect is zero. And so what this study is showing is that what you eat in a diet will affect what your resting energy expenditure will be, and the fewer calories you eat, the lower your resting, the higher your resting energy expenditure is, and therefore the easier it is to both lose weight and also to keep weight off. Well, that was one weight loss study, and the other weight loss study was in a much lower carbohydrate diet. That first one was 45% carbohydrate, so it was essentially a moderate diet: 45% carbohydrate, 40% fat, and I think 45% carbohydrate, 45% fat, and anyway, 45, 35, and 25, I think, or something like that. Anyway, so the second study was really a ketogenic level, so that was 7.5% energy intake of carbohydrates, and that study was, as I say, still unpublished. It's also funded by the Arnold Foundation, but they were able to take, Ludwig's group was able to take those two studies and determine that I should probably show the slides up locally so that I can follow along.
Actually,
here.
Richard, I've just got them here on my screen. I was just getting them sorted out while you're talking there. So let me just share my screen, and I think you should be able to see them here. There we go. So if you tell me forwards and backwards, and I will do that for you.
Go back for a bit. Okay, right there. So that was the Framingham State Food Study. It's a feeding study. They gave these people 60% of estimated required calories for 10 weeks to get them to lose weight. And then they did a bunch of other things with putting them on maintenance diets with different amounts of carbohydrates, and to get their outcome of the change in resting energy expenditure. But the important thing about this study is it was they did got them to lose weight on a caloric restriction over 10 weeks. And so they were able to use this information. And if you go to the next slide, please. And then this is the second one. This is the more ketogenic one. Next slide, please.
Okay.
Okay. So what they were able to do is they looked at a whole bunch. They looked at firstly the lean mass and the fat mass of these subjects and the change in that as they lost weight. So the question when you're trying to lose weight is, do you want to lose lean mass or do you want to lose fat mass? Obviously, you want to lose fat mass and retain as much lean mass as possible. So the interesting question, or the scientific question that they're asking is, what can we look at in these people to try and predict whether they will lose more lean mass or lose more fat mass? Is there any biomarker that we could use? They were able to find that if you look at the amount of insulin that they produce 30 minutes after a meal, then that is significantly associated with whether they're burning more fat or more lean tissue for energy. And so in one example, so for example, the FB4, which is the Framingham, Boston, Bloomington and Baylor study, where people got 60% of calories for 14 weeks, they had a lesser decrease in fat mass of 0.5% for every 100 micro IU of per mil of insulin increment. So, and it was extremely significant. So what that is essentially doing is it's giving us a formula to say, well, if your insulin 30 minutes after a meal is this, we can predict that we can predict the ratio of fat mass loss to lean mass loss. Next slide, please. Okay, so the question is really, why does insulin do this? What is it about insulin that decreases the amount of fat loss during weight loss and increases the amount of lean loss during weight loss? So no, that's okay. We can go on to this one. So this is a simplistic look at how insulin controls whether we're burning fat or we're burning glucose. And I know the question that we're going to answer is, why are some people burning fat and some people burning lean tissue? And we'll explain that hopefully. So in the low insulin state, you can see that insulin at the top center with a minus, that is a low insulin state, and insulin is not activating the insulin receptor there. And in that particular state, glucose can't enter the cell. This is a very simplistic way of looking at it. We're going to go in a bit more detail in a minute. And fatty acids do get into the cell. They go into the mitochondria. They get converted into acetyl-CoA, and then that gets turned into energy. And when you run out of fat coming through that pathway, then you start using amino acids. Okay, next slide. So that was the low insulin state. The high insulin state, now we've got a plus beside our insulin at the top. It's doing some magic with the insulin receptor, which we'll talk about in a minute. And that is allowing glucose to enter the cell. And then there's more magic that stops fatty acids from getting inside the cell. And now only glucose is getting into the mitochondria. Again, it's still being converted into acetyl-CoA to produce ATP. And then when you run out of glucose in a high insulin state, then you use amino acids anaerotically to generate energy. Next slide, please. Okay, so the question here is, I mean, this is a very simplistic way of looking at how insulin determines whether we're going to be either burning fat or burning glucose. But to understand, what we really need to understand is the insulin resistant and hyperinsulinemic state. So we need to go into a little bit more detail here. Next, please. Yes. Okay, so this again was our simplistic way of looking at low insulin, but the low insulin state. But fatty acids don't go flying into the mitochondria like that. They don't diffuse straight into the cell. No, that's good. Stay there.
Yeah. Okay.
Yeah. Stay there. Okay. So
I meant to do that.
Yeah. So there's actually a transporter involved called the carnitine shuttle, and that's actually going to get fatty acids, long-chain fatty acids, into the mitochondria. They can't naturally diffuse across that membrane. So if that transporter combination isn't working, then fatty acids can't get into the membrane, past the outer and inner mitochondrial membranes. And the other thing that's important here is, go back.
Sorry.
So that's okay. Another thing that's important here is that there's that transporter is rate limited, so it can only transport fatty acids at a certain rate, and above that rate, what happens to those fatty acids? Well, they don't slosh around inside the aqueous environment of the cytosol because they're fats, and fats and water don't mix. So they tend to form micelles. We know these as lipid droplets, and they're in all of our cells. They're a buffer of energy for when fatty acids aren't coming in from the blood supply, but you can still burn fatty acids in the mitochondria. The other thing about another nuance of the low insulin state is, in the low insulin state, that transporter for glucose doesn't sit at the membrane boundary, at the plasma membrane boundary. It's deep inside the cytosol of the cell, and it only comes up when there's an insulin signal. So that's what's actually stopping glucose from getting into the environment. Other than that, once you can't burn any more fatty acids, again, you burn amino acids. So the choice of the cell to utilize to draw down lean tissue in order to make energy is really a last-ditch effort when it doesn't have more available fatty acids or glucose. And the other thing is that we can also export these fatty acids out of the cell from one cell that has too many to one that doesn't have enough. And so the export of fatty acids is happening all the time. This is a cycle that's happening all the time. Okay, so back to the previous one. So that was the low insulin state. This is the high insulin state. When you remember, with this, the simplistic way of looking at this is fats can't get into the cell, glucose can get into the cell, but next slide, it's a little bit more complicated than that. So let's start this from the beginning. We've got insulin going up, so we have a green plus by insulin. It's triggered the insulin receptor, and the insulin receptor does a whole bunch of things. But two things that are important. One is telling GLUT4, that transporter, stop hiding in the inside of the cell and get yourself to the plasma membrane, because I want you to carry some glucose from outside the cell to inside the cell. And then the other thing that's happening is that the insulin receptor, when given a signal of insulin, also promotes the transcription of an enzyme. So it controls the transcription of your DNA into the protein for that enzyme called acetyl-CoA carboxylase. And acetyl-CoA carboxylase, the other thing that insulin also does, the insulin signaling does, is it turns that enzyme on. So, okay, what does acetyl-CoA carboxylase do? It converts acetyl-CoA, previous one.
Sorry, it just does by itself there.
No, that's okay.
It's got mind of its own.
Yes. And the other thing I should mention here is that not all acetyl-CoA, this is the global currency that both fats and glucose are being converted into. Not all acetyl-CoA gets turned into energy. So some of there's a shuttle that runs all the time inside the mitochondria that shares acetyl-CoA from inside the mitochondria to outside the mitochondria, because it's a useful molecule for building cholesterol and fatty acids and all other kinds of things. So so there's always acetyl-CoA available inside the cytosol, and it's coming out of the energy process. And the interesting thing here is when insulin turns on that ACC enzyme, acetyl-CoA carboxylase, that is the first step for taking acetyl-CoA and making new fat. So that's the process, that little square box there. Just go back to the previous slide. So there, that little box there with ACC, new lipid synthesis. That's where the cell, when given the signal of high insulin, is making new fats, and that process of making new fats inhibits the carnitine shuttle, so no longer can you get fatty acids inside the mitochondria. So that's the process, that's the switch of where it's being turned off. And of course, if you can't, if there's no sink for fatty acids, if you've turned off the ability to transport the fatty acids into the mitochondria to turn them into energy. Now you've got lipids building up in the cytosol, and they get more lipid droplets and more export of fatty acids from the cell. Next slide, please. Okay, now I think we're finally ready for the insulin resistant state. Now, the insulin resistant state, we start off really with hyperinsulinemia, and it's not just high insulin; it's Snoop Dogg high insulin. I mean, it's literally 2 to 3 times the amount of insulin, and also for hours rather than minutes after a meal in the insulin resistant state. So let's see how we get to that state. One important thing here is that if you're making lipid droplets compete with GLUT4, the transporter of glucose, for a membrane fusion protein called SNAP23. So if you make more lipid droplets and larger lipid droplets, there's fewer of these membrane fusion proteins for GLUT4. So when the insulin signal comes in, it says to the GLUT4, okay, get yourself to the plasma membrane to grab some glucose. It can't fuse with the membrane and get through to the other side. And that's the basis of insulin resistance. You have to keep pumping up the insulin signal just to get these GLUT4s to come up. And because we're in a relationship with the pancreas that's tasting the blood glucose level and delivering insulin in response, that insulin signal just keeps going up and up. Interesting thing here is that insulin resistance is downstream from the insulin receptor. So you might send it 2 insulins, but only one gets through to the GLUT4 because GLUT4 just can't get to the membrane. But you're still getting 2 to lipid synthesis. And so what happens here is that you're overdriving the generation of new lipids, and you're creating new lipid droplets, and you're also stomping on the transporter for getting lipids getting fatty acids into the mitochondria to be utilized for energy. And now you can't use fat or glucose and can't get them into the mitochondria. And that's the perfect situation for drawing down some of your lean tissue to waste for energy. And this is in the perverse situation where, the person's circulation is absolutely full of fat and glucose, and their mitochondria are unable to utilize it for energy. So that's really the explanation for why, depending on how high your insulin, if you're hyperinsulinemic with insulin resistance, then you will have higher insulin for longer after a meal, and you could be 1, 2, or 3 units of 100 micro IU increase compared to controls. And so that is why you're more likely to waste amino acids for energy, and less likely to use body fat.
Wow.
So that's my presentation, and I'm sorry for it being so dense, but I was a little bit flustered, but I'm open to any questions.
Wow. Wow, blown away. Can I just say though, it made me think. I love the way we started with the macro from Amber, and now we're going to the micro to see actually the human evolution story, and now actually how our bodies work. And the thing I think is really cruel is that I think sometimes, I don't know if you agree with me, that we speak to each other in the low carb community, and so we kind of get used to these ideas. And then when you speak to somebody outside, and I spoke to, as you know, Richard, somebody recently who is quite a famous scientist, and he literally said, "It's a calorie, it's all physics. You can't get away from that. It's energy in, energy out, nothing else." And I said, "What about hormones?" No, there's epidemiology to prove this point. Please don't kick off, Nina. And he said, and that is his argument. And I was like, that's a dominant still now argument made. And you just literally said that if your insulin's too high, you literally cannot access your fat to burn it.
It's not a calorie. It's just fat is not a calorie. If your insulin is stomped on your carnitine shuttle, fat is not a calorie. It is just a metabolically inert molecule that you have to store somewhere. You look at people who are insulin resistant. This is a mindset change. Once you realize these people can't burn fatty acids, and it's just junk that they can't utilize, it entirely changes the way you look at type 2 diabetes.
Yeah, completely. And also all the other metabolic syndrome symptoms you see, it's global, isn't it? Nina, I will stop hogging, I promise. What would you like to say?
No. I just think it's fascinating to connect these ideas back to what Amber is talking about. The idea that early humans hunted large animals precisely for fat, for energy. And here we are in the modern day world where we, due to hyperinsulinemia, cannot use fat for energy, is what you're suggesting, right? I mean, how ironic is that? You're eating the very thing that you need for energy, and you can't use any of it. It just goes straight to fat cells.
Yeah, it also ties in with Cynthia's talk as well, because you need that downtime of low insulin to be able to release the spigot. So it's we tend to obsess about the source of fat in the diet, fat on my belly that I'm going to utilize, but we don't think about the sink, and the source and the sink are both coupled. So one example, what I was showing here is how the sink gets clogged up, and you know, if you have a bathtub that's full, and you have a hand washer in the tub, and you add more water, it doesn't matter how much you turn the water on or off, you're still going to overflow until you pull the hand washer out of the sink. So yeah.
Did you want to say something, Cynthia?
I did. I mean, I'm the clinician that now knows better. I think to Jason Fung talking about the kind of old paradigm of calories in, calories out, CICO has so largely been disproven. And I'm so very grateful for the scientists, the biochemists, like some of the people on this panel that are talking about the research about hormones and hormone imbalances that are driving our inability to lose weight or our ability to put on weight. I think it's really important. And, you know, if there's anyone that's watching this podcast or watching it live or watching the replay, we as clinicians need to learn and do better for our patients, because this is a critical understanding. I still argue with other healthcare professionals online about the CICO, crap that a lot of people still perpetuate. So thank you, Amber and Richard and Nina, for, you know, helping to educate all of us and, you know, teaching that, you know, the old dogs can learn new tricks. And so I think it's really important that we are evolving as clinicians and not staying so constrained by antiquated dogma like so many of my peers still do.
Before I go to you, Amber, because I know you wanted to say something too, I just want to say the thing I noticed in somebody I spoke to recently was that actually what Richard's gone into there in depth and detail, I don't think he and other people have looked at. They start with a paradigm and then they build on the paradigm without realizing the actual premise itself is the foundation is wrong, so everything else is wrong. And if they went back to the beginning, they'd realize that, hang on a minute, maybe 10 years of their career has been based on something that's not right. And what I find really interesting is I always thought that science was pretty straightforward, that it was a matter of truth and it's right or wrong. And you literally just discover, take off, you know, the cover and decide what you see is what's there. And that's the evidence. But it seems like these paradigms dictate so much. And so what really should change in the matter of a 15-minute talk takes generations, and I find that really shocking. That's humans for you, isn't it? Amber, I said I'd go to you next. Over to you.
Thanks. Yeah, I was just thinking that this has implications for protein and fat ratios as well, especially in people who have high basal insulin, because in those people, if the more protein and the less fat that you have in a particular meal, the higher your insulin response is going to be to that. And then, well, then I guess it comes around to it's going to increase your protein need. So eating higher protein kind of begets higher protein need.
Mm-hmm. Well, Gannon and Nuttall discovered that type 2 diabetics produce almost as much insulin response to a meal of solely protein as they do from solely glucose, and 2.5 times as much from glucose plus protein. So 50 grams of glucose, there's a certain amount, let's call it 100. 50 grams of protein, it's 90. 50 grams of a mix, 25 grams each of glucose and protein, and it's 250. So the body is amazing. It's magic.
Wow.
But yeah, the thing about you're right there about protein being a challenge for type 2 diabetics. One interesting thing I found when researching this, that mechanism, that inhibition of palmitoyl transferase, which is the carnitine shuttle, the inhibition. So if you make new fat, that stops you from getting fat into the mitochondria. That inhibition is different for different people. And for example, in diabetic rats, that the rate constant of inhibition is 50 times that in a non-diabetic rat, which indicates that a diabetic rat is making 50 times more fat than a non-diabetic rat on the same rat chair, which explains some of the challenges of metabolic syndrome.
Wow. You know, we had, I don't know why you'd know this. We had the half marathon in Cambridge at the weekend, and I saw at these checkpoints, they had all the sugar pouches that people were, ingesting as they went through, because
Blame
clearly,
Tim Noakes.
yeah, I do. And the fact that, obviously evolution has made us to only have a very small fuel tank that we have to completely replenish every, like few miles or whatever. Otherwise it's gone completely. And yet at the same time, and I, by the way, I'm not fit enough to run a half marathon, so I'm definitely not having a go at anybody. But there were unfortunately some runners who had a lot of fat on them and they're not accessing it because exactly what you just said. They're eating the sugar to try and make the glycogen or whatever, and their fat stores are sat there. All that energy should actually, if they fasted, they could live off like a month or more easily, and they're just not accessing it. And it's an injustice because they don't know. That's what annoys me, because the medical profession, as Cynthia said, it doesn't understand itself. So of course, what chance is a layperson like myself, if they don't know about these sorts of conversations, how would we know?
I know the scientist. Yeah. I was just going to say, I know the scientist you went to talk to, and I know where he's coming from, and it's understandable because it's a post hoc logic fallacy. If you look at a group of people who are type 2 diabetic and another group that aren't type 2 diabetic, you'll quickly see something that's fairly obvious. The people who are type 2 diabetic are mostly overweight. And if you look at when their blood sugars go out of control, it occurs 5 years after they really put on the pounds. And so it's a post hoc ergo propter hoc logic fallacy. They got high glucose after they got fat, therefore getting fat causes diabetes. But 5 years before getting fat, their insulin, they started to develop the Kraft insulin resistance patterns, and so it was a third factor that is underpinning this, that's causing it. So let's say you've observed two groups of people, and you've made the obvious conclusion, being fat causes type 2 diabetes, and somebody says to you, well, there are thin people who have type 2 diabetes, and there are fat people who don't get type 2 diabetes, and so rather than saying maybe the explanation isn't correct, people say, well, there must be something unusual about people, a personal fat threshold that each individual has that's magical and determines at what point their fat fills up, and then everything all starts to go downhill and they become diabetic. And they've gone down this path, and it's a dead end, and because it fails to explain all sorts of things like, why do people's triglycerides increase? Triglycerides in circulation increase all the way along the pathway from starting to have high insulin to finally getting diabetic. So unfortunately, it has grown the way it has, and we now have a very large ship that won't turn around on a dime, unfortunately.
Wow. Yeah, you're right. And we don't have time, 10, 20 years. That's millions of people affected by that paradigm, isn't it? That's wrong. Just before I go into our next speaker, before I, finish up on that topic, I was going to share some comments here as well. Nicola Howard said, it's all about nuance. We're trapped in a world that demands simple and trite answers. I read in a certain book somewhere, I think I know exactly which one it was. It's by you, Nina, and where you quote this idea that, why is it called fat if it doesn't make you fat? And it's such a simple answer that people think, because that's what they get told. And of course, it's never that straightforward, is it, when you understand how the body works? Then we have Faye here, Faye Louise, who says, fantastic, Richard. Thank you for the detailed depth of pathways, which is fantastic. And by the way, this video will be available after this. I know that most of my audience will be asleep right now because they're sensible. So if you do come back tomorrow, you'll be able to find it when you wake up. Sorry, you'll find it all there with the pathway information. We'll do links to all of that so you can see all of those charts and whatnot for yourself as well. And then Susie Edge said, who I know very well as well, it's so hard to get this idea across, the seco-persistent idea, which of course is so true. So excellent. Thank you so much for that, Richard. That's fantastic. And just to let everyone know who's following now, if you haven't followed us on Twitter, please do. I've been trying to tweet at the same time as doing everything else. And if you look there, there's actually a hashtag that I've used, which is Big Fat Combo. I know it's classy, isn't it? So if you have a look for that, you can actually see I've tweeted everyone on the panel tonight, and I'm sure their articles and stuff will be there very soon as well. But I'm going to move on now. And that's to our last speaker this evening, this morning or this afternoon. And that, of course, is Nina Teicholz. She's a New York Times bestselling author with her book, The Big Fat Surprise. Nina is a science journalist, speaker and researcher. She's also the director of the Nutrition Coalition, a non-profit group that promotes evidence-based nutrition policy. Welcome, Nina, to the show.
Hi, thank you so much.
It's great to have you
be here.
And I love the fact that you're so beautifully lit up this evening. I don't know if you've invested in
a joke. Yeah, I invested in a light ring, but no stand, so it's like teetering on it with a bunch of books around it, so it might fall over at any moment.
As long as you don't say, nobody would know.
Yeah, and it doesn't matter because now I'm speaking to all the insomniacs in England, at least, or in Europe.
Okay, I was going to make you the big screen, and then I just want to talk about your paper today. So you are talking about this paper here, which is Saturated Fats and Health, a reassessment and proposal for food-based recommendations. Right. I can pronounce everything in that one, unlike Amber's, so there you go. I feel very happy. So yes, over to you, Nina Teicholz.
Okay, thank you. Well, I'm presenting this paper, which is... it was published in the Journal of the American College of Cardiology, which is a very prestigious journal that cardiologists read and others. And it was called a state-of-the-art review on saturated fats. And I think what is extremely notable about this paper is that the authors include 4 professors who had been members of the U.S. Dietary Guidelines Advisory Committee. So those are the people that actually make the nutrition, our national nutrition policy, and there's a new committee every 5 years to review the latest evidence. And these are people who are saying, you know, we got it wrong on saturated fats. So it's a very sort of high level paper in a very an excellent journal, and it came out in 2020. And at the end of the year, the editor-in-chief of that journal selected it as one of the top 5 papers of that year. So it really is quite a notable paper, because although there have been numerous scholars really around the world, journalists like myself in my book, and others saying that they don't believe the evidence for saturated fats to support continued caps on saturated fats. They don't believe the evidence has ever been strong. This is a really high level paper coming to that same conclusion. And I'll just read you the exact conclusion that it comes to, which is, taken together, the evidence from both cohort studies, so that's epidemiological studies that show association, and randomized trials, which is the more rigorous kind of evidence, does not support the assertion that further restriction of dietary saturated fat will reduce clinical events, meaning heart attacks or any kind of cardiovascular, any kind of event due to coronary heart disease. So that's pretty striking coming from this group of people. And so I want to explain, like, I know that many people know this, but I sort of just wanted to explain what is the history coming up to this paper. We know this is an important paper that came out in 2020. And why is it that we still have, in every country in the world, caps on saturated fats, if there is such a paper at such a high level? So as many people know, the idea that saturated fats and dietary cholesterol cause heart disease goes back to Ansel Keys, a physiologist from the University of Minnesota who proposed, he was the main academic to propose this in the late 1950s. And then the American Heart Association was the first organization anywhere in the world to sort of formalize this hypothesis and tell the public that the best way to avoid getting heart disease was to avoid foods that contain saturated fat and dietary cholesterol, which on a practical level meant cutting back on meat and cheese and butter and coconut oil, or if you live in Asian countries or came from an Asian background, which is the primary fat used in some of those countries. So, and so what happened was that, even at the time this Heart Association recommendation came out in 1961, there was very little evidence for this idea. And so what happened is that governments all over the world undertook randomized controlled clinical trials to test this idea. They literally put thousands of people. It's thought that more than 75,000 people were tested in the 1960s and 70s on this idea that the question, does saturated fat and cholesterol cause heart disease? So they literally would take groups of people. Many of these experiments happened in mental institutions, the kind of experiment you can't do today because it's considered unethical. But they were ideal because they could control what people were eating inside of an institutional setting that was relatively stable, not perfectly stable in some of these experiments. And they gave their experimental group would have soy-filled cheese, soy-filled milk, whatever the equivalent of the the Impossible Burger was in those days, and the control group would get the regular diet, whatever they consider the regular diet: real meat, real cheese, real milk. So, and these studies, on the whole, did not support the diet heart hypothesis, which is to say that they really did not show that the people on the soy-filled cheese and soy-filled milk and all of that, they did not suffer reduced rates of death from heart disease. I mean, this was a pretty universal finding, but because there was such a strong bias in favor of the diet heart hypothesis, most of those findings were sort of ignored or suppressed or misinterpreted, or they just kind of got twisted in, and they really didn't become, they just sort of fell out of the scientific literature and were not cited. This is something that I write about in my book if you want to read more about it. But I think, so what has happened is that since 2010, so really the last decade, academics all over the world have gone back to those original clinical trials, those sort of lost clinical trials. And that is because they found out about them due to my work, due to the work of Gary Taubes and others. The first set of papers came out by Ronald M. Krauss now at UCSF. And these papers kind of dug up the clinical trial evidence and said, well, what is the story here? We have so much really good, rigorous data. And what were the findings? And what they have found, there's now more than 20 review papers looking at the clinical trial evidence, also the observational or epidemiological data. And these papers have nearly universally concluded that, they've universally concluded that saturated fats have no effect on cardiovascular mortality. That means dying from heart disease, or total mortality. That means dying from any cause. And most of the papers have concluded that that saturated fats have no effect on cardiovascular events, which are like heart attacks, myocardial infarction. So this evidence from the last decade, this kind of reevaluation of the evidence on saturated fats, has really been a sea change in our understanding on saturated fats. It's a huge body of review papers that have gone back and reconsidered saturated fats. And they've looked at, some of the questions that people have about saturated fats. Well, don't saturated fats raise your LDL cholesterol, which is the so-called, bad cholesterol, which can be disputed. But this paper, deals with that, that issue. Well, and what should we think about the fact that saturated fat raises LDL cholesterol? And basically, one of the answers, without going into too much detail right now, but one of the answers is that lowering your LDL cholesterol through diet, by changing your food, has not been shown has reliably not been shown to reduce your risk of cardiovascular disease, compared to, and this is a comparison that's been made, where it does seem to be that taking a statin, this is also disputed, but it's generally accepted that taking a statin or taking some kind of medication that lowers your LDL will lower your rate of cardiovascular events. But that cannot be translated into lowering your LDL using a food intervention, a dietary intervention. And it also turns out that saturated fat lowers the kind of LDL. LDL comes in various varieties. There are different types of LDL, and saturated fat does not negatively affect the type of LDL that is thought to be most atherogenic, meaning most likely to cause heart disease. So I think that, and another thing that I love about this paper is that it acknowledges sort of the historical role that foods containing saturated fats have had in human evolution. That is something that, in general, the nutritional community is sort of averse to looking, or the high-level nutrition community is averse to looking at sort of historically how humans have evolved, exactly the kind of evidence that Amber talked about. But this paper really does talk about that, which is wonderful to see, because I think it's just so highly relevant. Anyway, let me address this just one question, which is, why is it that dietary guidelines around the world still have these caps on saturated fats? This paper came out. It was hand delivered to the dietary guideline expert committee that was reviewing all the data for the most current set of guidelines. This paper was submitted as a formal public comment. The authors of this paper went to talk to high-level officials in the government agencies that oversee the dietary guidelines, and it was ignored. And in fact, all those 20 papers reviewing the data on saturated fats from the last decade were all ignored. And so what we have is a situation where we have a very entrenched idea, a kind of dogma about saturated fats. It's been around for 60 years. And even though there's a tremendous amount of science to the contrary now, I think that we're basically in a situation where the science is it's until it can be actually heard by the proper officials in government agencies. Even when people come knocking on the door and hand it to them, it's still the case that that we're just living with these antiquated, outdated dietary guidelines that are actively causing harm. I think it's fair to say. And just to relate that statement to Richard's presentation, it's so clear, right, that in his presentation, that people with metabolic disease, in that case type 2 diabetes, they need a different diet. Their body works differently than people who are of kind of regular good health. And so we cannot continue to have a set of dietary guidelines that's a one size fits all, and that is based on outdated science. So I'll just mention one final thing, which is there's another paper that came out in the journal Nutrients, where I'm an author, and it includes a number of other authors, some of whom were participated in this JACC paper that I just talked about. And that paper actually takes an in-depth look at all the science that this most recent dietary guideline advisory committee did look into. It lists all the studies and shows that their own review, 85% of the studies did not find an association between saturated fat and any kind of negative outcome for heart disease. And yet they still concluded that saturated fat causes heart disease. So there were, I think it's fair to say, sort of betraying their own evidence. And it turns out that saturated fats has consistently shown to be associated with a lower rate of stroke. In other words, eating more saturated fats would be preventative against stroke. So I think we're a little bit in the twilight zone when it comes to saturated fats, but the evidence continues to mount, and scientists continue to speak out, I think, on the other side of the issue. And so I hope that we'll eventually see guidelines that reflect the science. And that's all.
Wow. That's all as if that's nothing. That's incredible. Okay, there you go. Blown away there. I just want to share something because I was reading your paper today and I've done my highlighting. And
Oh, good for you.
this part in particular I like because it relates to the UK. So 195,658 participants were on a 10.6-year study. No evidence of saturated fat causing cardiovascular disease, but polyunsaturated fat, there was a link to cardiovascular disease. Now, that's amazing evidence. I mean, take that paradigm. What are you going to say to that? So my question, I suppose, is with that kind of evidence, why on earth is it being ignored? That I know you raised that, but I just want to ask, what do you think is going on?
Well, the answer to that is complicated. I think there's various forces at work here. There are people who've devoted their whole careers to showing that saturated fat is bad for health. In fact, the head of the subcommittee reviewing saturated fats for this most recent dietary guidelines was a professor named Linda Van Horn, who's literally spent the last 50 years of her life trying to show that saturated fats are bad for health, and worked with Jeremiah Stamler, who was a close colleague of Ansel Keys. So what you have here are, really strong intellectual conflicts of interest. And then I think it's undeniable that the food industry plays a role here because polyunsaturated vegetable oils, which is what you eat when you replace saturated fats, they're sort of the backbone of the food industry, and they're huge companies involved here. We're talking Bunge, ADM, Monsanto, Unilever, Cargill. I mean, they're just gigantic companies that rely on these cheap fats. And I know from some of my reporting that these companies are fund nutrition scientists, have close relationships with the government U.S. agencies that are involved in the guidelines. So I think that there's a lot of vested interest involved in maintaining the existing status quo.
Yeah. And that's exactly it. The status quo is a very convenient thing, like you say. I totally agree. Would anyone else like to come in there to either make a point or ask Nina a question or anything at all about her paper? Go for it, Richard.
I was at a conference in Zurich. Nina was there as well. It was an awesome conference. And I remember the editor of the BMJ. So there was a presentation by Darius Mozaffarian and Salim Youssef, and Salim was talking about the Pure study, which showed that there was no association between saturated fat and cardiovascular disease. And I asked him a question about why is that still the dogma? And Darius Mozaffarian jumped in front of the bullet, and he said there's been 19 randomized clinical trials testing this hypothesis, and it's never turned out. Nobody should be shocked that saturated fat has no association with cardiovascular disease incidence. And Fiona Godley, the editor of the British Medical Journal, said after that, she said, well, I think we can come out of this conference with two clear agreements that diabetes can be reversed, which is now dogma, and that saturated fat doesn't cause heart disease. And there's still nothing happened. This was 3 years ago, Nina. It was just...
Yeah. I don't just say that. I mean, she actually said, I think we owe a big apology. We got it wrong on saturated fats. She was quite, it was a shocking comment, but I think there's just a lot at stake. I mean, it's interesting, Dariush Mozaffarian himself has different conflicting interests. He was on the scientific advisory board of Unilever, which was then the largest manufacturer of polyunsaturated vegetable oils in the world. I mean, there's just a lot of vested interests, and I could talk about this all night. I'm not going to do that, but there are enormous interests, like the animal rights groups, who are very, very well funded, and they don't want people switching to saturated. They don't want to lift the caps on saturated fats because that is essentially a sort of rate limiting factor on how much animal foods people eat. So they don't want them eating more dairy, and they don't want them eating more meat. And that's a very active group. And we all know we live in a sort of a world that it favors plant-based foods now heavily. So anything that is about going back to animal fats is, there's just many forces lined up against it. So I don't know.
What you're saying
I
is
just don't
it's
know.
religion.
It's religion and financial interests, which are just,
Yeah,
I mean,
the worst.
it's, I don't know. I don't know where, I mean, I think it's going to be hard to change that because, I mean, who are the lobbies that would favor saturated fats? I mean, they're, the meat and dairy industries, which
Cattlemen.
are not too popular. So it's a really difficult thing. But I think, the tragedy is just that I want to say, and the health tragedy is that people are not able to eat the foods that contain all the micronutrients that are needed for human. And this is especially tragic because our dietary guidelines controls what goes in school lunches and feeding programs for women and their infant children and elderly and all of this. So if they're not allowed, if there's a 10% cap on saturated fats, which there is now, 10% of calories, so they're just not allowed. They can't get regular cheese. They can't get they definitely can't get meat or very little of it. And red meat especially as a source, as you all know, of bioavailable iron and other really important nutrients that are hard to get elsewhere, or you can't get them, absorb them as readily from plants. So it is like an active tragedy that we cannot get our guidelines to reflect the science, because it really translates into poorer diets for humans.
It's going to be fascinating with the sort of view of history, I think, because history will look back on this generation. It will have its say, just like we have our say in previous generations, and it'd be very interesting to see the kind of the impact that happens on society with the plant-based diet. So, I just think about the mental health potential problems that could be in the future. We don't know about the impact of things like cancer. Certainly, there's a link with sugar, isn't there, with things like cancer? There's so many things that could be the start of another pandemic of ill health that we don't even know about yet. And history looking back on this will be very interesting.
Well, maybe we'll just all be too demented at that point to even know what's gone wrong.
We won't be, especially not Amber. So, okay. Yeah. I was going to go to Amber now because I think you want to come in there and ask something.
Oh, yes. Well, the point I was going to raise is actually exactly the one that Nina herself raised, which is that even though plants can be a source of saturated fat, the only remaining nutritional argument against eating meat is the saturated fat one, and if they lift that, then there's no more reason not to eat meat than that is nutritional that they can fall back on, and I think that may be a primary sticking point.
I think you're right, Amber. I mean, I think, I think you're right. And as the science for saturated fats started crumbling, you could see the rise of other reasons not to eat meat, which we won't get into here. But, for a long time, it's been saturated fats. So it's going to be a hard one to crack.
Yeah, for sure.
But okay. I want to say to end on a positive note, that the reason I shared this paper is that it's the Journal of the American College of Cardiologists. Those are cardiologists. They were the first people to say, don't eat saturated fat for heart disease. And if they start to be convinced and understand that changes in the diet, it's not like they can still prescribe pharmaceuticals, but the diet part of it can be a different story. I think that it's possible that change, that community couldn't be a force for positive change.
Definitely. Thank you so much for that, Nina. No, that's fantastic. And there's actually something here, and I know we've got some comments to go to in a moment, but just about the articles themselves, there's been real strong links between them, haven't there? But very different angles, which I think is really important. And if there's anything that I think I've noticed, and okay, just for those people who are watching right now, you don't know who I am because I'm not in the same league as these people. I'm the layperson who asks the questions because I don't know and I want to find out. And I think when I speak to specialists, sometimes they very much look at their one area. And like I said before, they have the paradigm which they base everything else on. And I think it's really important to hear these different angles. So you've got the evolutionary angle there, the actual case studies into fat not being dangerous for us, the way the cell works, the way the microbiome works. There's so many different angles, and tying it together, you can definitely see a picture which is much bigger than just the sum of its parts. So, yeah, thank you so much for that paper. And I'm just going to go to the comments now, because I appreciate you've all been here a long time and I don't want to keep you all night and all morning. So let's just go to this one. And I'm just going to direct this to initially you, Richard. And I think Cynthia could also take this one on. So Donna here says, "Hi, Richard. I'm interested in active post-menopausal women and how to retain lean mass and bone health. Are you aware of studies looking at estrogen and how cells use it and the transporters?" I'll let you have a go at that first.
I don't know a lot about the movements of estrogen during menopause, so I'm going to hand that over to Cynthia. But I do know that estrogen is absolutely essential for growth, maturation, and remodeling of bone. There are receptors for estrogen on osteoclasts, osteoblasts, and so I mean it is critical. And I would suggest that you probably want to talk to a physician who's able to analyze your estrogen levels, so that you know if they're indicated, if replacement therapy is indicated, and somebody who's willing to interpret the results and not just knee-jerk respond with, well, you got to have hormone replacement, because maybe you don't need it. But I'm not an expert, so I'll hand over to Cynthia, who's muted.
Yeah.
Sorry, I was getting ready to speak and realized I was still muted. Thank you, Richard. Yes, I think this is an area where there's a lot of misinformation for many women in perimenopause and menopause. I think it's really critically important for women to understand that one of the ways that you are able to maintain muscle mass is you have to have proper stimulus. So you need to lift, you need to do strength training, and you also need to consume adequate animal-based protein. And I talk very openly about this. And Dr. Gabrielle Line is a great resource. She's a female physician here in the US. She talks a lot about muscle-centric medicine, and she's kind of leading the charge. And I'm definitely one of her supporters. And so, you know, part of it is the adequate intake of protein, provided also an adequate stimulus on the muscle with strength training. But for many, many women, we don't realize that our peak bone and muscle mass building is in our 20s and 30s. And sarcopenia, which is this muscle loss with aging, starts to accelerate after the age of 40. And so what you see is a lot of women reporting things like, it's harder for me to maintain muscle. I don't understand why this is happening. I'm told I'm osteopenic, meaning that my bones are weakened. And if you exist in a lower estrogen state most of your adult life, and a lot of women do because they're on synthetic oral contraceptives, IUDs, et cetera, that can actually cause an exacerbation of this kind of low estrogen state. So you're heading into menopause already in lower estradiol states. And to Richard's point, we do have estrogen receptors on muscle as well as bone. Also, the loss of testosterone can also exacerbate this. So I think you need a multi-pronged approach when you're trying to mitigate some of the hormonal fluctuations that occur at middle age. You do absolutely need serum, so blood testing. I do like utilizing a test called the Dutch, which looks at dried urine and oftentimes saliva, a multi-pronged effect. And I think it's also critically important that women not be fearful of utilizing hormonal therapy, because it can maintain muscle mass, it can maintain bone mass, it can also protect your brain and your heart. And there's a really excellent book by Dr. Lisa Mosconi, who's a Alzheimer's researcher, really looks at female brain health. She's based out of Cornell, and she has a book called The XX Brain. I encourage every woman to read that book. It changed my entire perspectives, not only personally, but professionally, about the need for consideration of hormonal replacement therapy. So I hope that's helpful.
Fantastic. Thank you so much for that answer. That's, well, yeah, the right panel for the right question. And fantastic. I just want to say, though, to everyone on the panel today, and also for those people who are watching, especially if you're like a diehard who stayed up with us through the night here in the UK, thank you so, so much. And I just want to say a massive thank you to my panel as well. I know people up and down the country right now are clapping and cheering. I can hear fireworks going off. Yeah, that's for you guys. So I just want to say a bit of BS there, sorry. I just want to say a massive thank you to all of you. It's been a really fantastic conversation. I just want to just give a quick plug, if I can, to the Public Health Collaboration here in the UK. If you don't know that charity in the UK based, please look them up. They're a fantastic charity who are actually trying to make a real difference, like some of the things we've heard about today, in doctor surgeries with people who are either type 2 diabetic, other metabolic syndrome conditions, and then they train up ambassadors to go in to talk to the doctors to try and make that change. So really important work they do. Thank you to everyone here. I wish you all now a very happy evening, morning, whatever you're up to. I'm going to bed, and I think we're done. So bye, everyone. See ya.
Great
Bye.
to spend time
Thank
with
you.
you.